Okay, now Sam,
Okay,
please.
great. Thank you, Hyson, for the introduction and for the invitation. Um, I guess the thing I want to say first is that I'm going to overview a couple different topics. So I'd appreciate if you have questions, ask them throughout. That sort of makes the most sense. But yeah, so I'm going to talk about some of my postdoctoral work today on model experimental systems of DNA liquids, like these DNA nanoparticles here that assemble into these dynamic and disordered meshworks that result in liquid droplets on the micron scale. So not only I'm going to talk about some near equilibrium dynamics and patterning, but also how we're thinking about driving these kinds of phase-separated systems out of equilibrium with enzymatic reactions. So I want to start with this sort of remarkable fact, at least to me as a physicist, I still find it very interesting that in our bodies right now, we have 10 to the 13 different cells, but all containing the same genetic information. So on the nanoscale, the information is the same, but via differentiation and other cellular processes, they can assemble and organize into these very hierarchical structures that can span length scales that allow living systems to do very complex tasks like force generation, chemical processing, and computation, which I may get to a little bit. So how does this all work? We all learn in our introductory biology class, the so-called central dogma of cellular biology, that the code of cellular function is written in the DNA. That DNA is then transcribed into RNA, that is then translated into a protein, amino acid sequence that folds and then performs a particular function. And I guess I want to ask quickly, can you see my cursor? Is that yes, okay, great. Okay, so this sort of pathway encodes one function, but let's say you want to do perform more than one function given a single cell. You might imagine that the reaction network looks something like this, where you have different genes encoding different RNAs that then determine different proteins. For E. coli, that's roughly 4,000 different genes in the genome. Humans have more, of course, but on the order of thousands, tens of thousands. But of course, it's not this simple. This is an overly simplified reaction network. Just 20% of the E. coli transcription network looks like this. So the nodes here are transcription factors or proteins that can then up or down regulate other genes in the genome via these arrows here. So it's not a simple reaction network like I showed on the left. Even then, the reaction network sort of perspective of the cell is not enough to explain some of the very complex behaviors that have been observed in biological systems. For example, reaction to fusion patterning is a very important one on a way that living systems can span length scales from the nanoscale up to the organism scale. So one example here is the so-called MIN system in E. coli that determines where the cells divide. There are also these animal skins that occur from reaction to fusion patterning, and also Drosophila morphogenesis here, where cells signal and the intracellular transport is what determines, for example, where these body segments form. But the one sort of physical principle I'm most interested in today, and one that's relatively recently discovered, is that of liquid-liquid phase separation that uses the physics of binodal coexistence, let's say, to organize biomolecules inside living systems. So here's one example on the left, which is more of a physical one, where these phase-separated droplets of beta-carotene in tropical bird feathers lead to these brilliant structural colors. That is a scattering effect, essentially, from the phase separation process. But the other one that's sort of maybe more interesting or more complex is that of genome regulation. Here is a picture of the nucleolus in vivo where there are phase-separated droplets in the living system. So it's not a homogeneous bag of biomolecules. They're separated into these droplet-like structures with this hierarchical organization. Okay, and so this kind of sort of organization from a phase separation perspective has inspired the engineering of these sort of compartments that lack membranes, sorry, that have a specific biochemical composition. And so from an engineering perspective here, this is what you would call a synthetic cell, so-called engineering synthetic biology from the bottom. So the typical approach, the top-down approach, is one of live cell engineering, where, say, you want to perform a treatment on a patient, you take some living cells out of them, then you modify these cells and maybe you grow them back up after you modify them, and then you infuse them back into the patient to achieve a desired effect. But of course, this kind of process is costly and invasive and has limited scalability from an engineering perspective. The other way you can think to do it is to essentially cut out this bottom step here and assemble the cells from the bottom up from biological or biologically compatible synthetic components with a programmed biochemical function. And so I just want to point out here, relative to the sort of typical approaches of doing these, of making these synthetic cells, which depended on lipid vesicles and microfluidic processing techniques, phase separation can give you a sort of a scalable approach to self-assemble the compartments just using the physics of phase separation. And so there's, of course, a clear application perspective here, but I would argue that there's also a very important fundamental motivation here. That is, if we can build these compartments from the bottom up and we can quantify them accurately, we can learn life's molecular and mesoscale processes. So this is sort of as close to a mission statement of my work, if there possibly is one. Okay, so just an overview of what I'm going to tell you about today. On the left here, which I would call near equilibrium patterning and condensation dynamics as a result of thermal quench into the coexistence regime. And then on the right here, one project about condensation dynamics with enzymatic reactions. Okay, so this is the model system we're going to use to investigate these processes. So here it is a self-assembled DNA particle that we designed these 4 different single strands of DNA to assemble into this junction-like structure. And at the end of each junction is a sticky end that has a complementary sequence. Here, these are palindromes, so every end can bind to every other one. Below a given temperature, given this binding strength, they assemble into these meshworks on the nanoscale that then result in liquid droplets on something like the micron scale. I like this figure because it sort of emphasizes the hierarchical assembly of this, but of course, what is problematic is this image right here. This is not a 2-dimensional array of nanostars. It's a 3-dimensional liquid structure. And so I also want to show this simulation of the assembly of the DNA nanostar liquid, just to emphasize that this is a 3-dimensional network that is disordered. The binding between the sticky ends is very weak, just a few KT, so that the meshwork is dynamic. This is what leads to the liquid behavior. And also that the structure is very open. So the DNA here occupies only roughly 1% of the volume. And so really the benefit to this kind of model system is that we have complete control over the nanoscale design, the size of the particles, the number of arms, let's say, and the binding strength and sequence. And this controls the bulk liquid properties. So you make the binding strength stronger, you increase the viscosity substantially, and in other ways, you affect the surface tension and the density as well.
Can I ask a quick question over here?
Yes, of course.
So the size that you mentioned over here, this is the size of each individual nanostar. Is that correct?
Yes.
Sort of radius of gyration or something, presumably.
Yeah, so here we sort of think of the arms as infinitely stiff because the persistence length of double-stranded DNA is around 50 nanometers, and these are much smaller than that. Yeah.
And do you have a sense for how the number of arms versus the size controls both the mesh size, so the pore size of this porous, very loose network, and also the entanglement length? Because presumably if you have many arms, then you start tangling up as well.
So the first point about entanglement is an interesting one. I would say there's no entanglement here. You can think of this kind of system not as like a polymeric system, more as like a pachycolloid in this limit where the arms are short relative to the persistence length of the DNA, right? And so essentially what you're seeing, if I can go back, sorry, is if you draw circles around the junctions of radius of the arm diameter, you will just see a packing of pachycolloids. And so actually one of the real successes of this kind of model system is they call this an empty liquid based on Francesco Sciortino's sort of pachycolloid theory, in which, if you have 2 plus epsilon sticky patches, you can get a liquid with zero density, sort of the idea.
I see. Okay. So then the pore size, the mesh size is also roughly the size of the units, the stars.
Yeah, that's right. That's right. And so we're typically thinking of these in the limit where the meshwork is fully connected. So of course, if you're near melting, that's not quite true, but it turns out very close to the melting temperature, fully connected liquids, but that are also dynamic. Yeah.
Thank you.
Okay. So the first thing I want to tell you about is sort of our typical experiment. This is what we do. We prepare the system such that it's a uniform homogeneous liquid of unbound nanostars above the melting temperature, let's say, and then we quench into the coexistence regime, and then droplets start to form in the movie here. So you can see droplets start to form, and then eventually, because the DNA is denser than the solution, they will sediment into the bottom, into these two-dimensional layers of droplets. And so the first thing I want to tell you about is the dynamics of condensation. So here, what determines the onset of droplet formation? Okay, so what we do is we do that kind of experiment we quench into the coexistence regime with what we would call a deep quench here, 6 degrees below melting. And at different nanostar concentrations, we just take movies, microscopy movies, and we can determine the onset time of condensation. So here, as on the left, there are no droplets formed, and then on the right, you start to see little flecks floating around before late times where you see larger droplets form. And so the important length scale here is the microscope's detection limit, which is just the point spread function given our microscopy setup. And for us here, this is about a micron. But for the qualitative behavior here is that for high nanostar concentration, droplets form quickly, but that at lower and lower concentration, it takes more and more time for droplets to grow to this micron size. And so we can plot that now, the delay time as a function of nanostar concentration. We see this nice power law behavior that is consistent with what you would expect for Brownian motion and coalescence mechanism. And so here, the average droplet size grows as t to the 1/3 with this prefactor here. And this prefactor has the bulk nanostar concentration in it. And so that agrees very nicely. And the other sort of quantitative thing to say here is that the surface tension of these droplets is very small. So processes like Ostwald ripening, which have the same exponent, are not relevant here. Essentially, the prefactor for the Brownian motion coalescence is 4 or 5 orders of magnitude larger. Okay, so this is great. We see a nice agreement with the scaling, but if you look at the prefactor here, the experimental growth rate is 2 orders of magnitude slower than what we would expect from the theory. And so essentially what we do is we turn to some simulation to try to model what's going on here. And because we think we have a good idea with the Brownian motion and coalescence, we perform particulate simulations where we model the nanostar droplets as just coalescing droplets that undergo Brownian motion with Stokesian diffusion. And so this is a 2-dimensional example of what you would see. You start out with very small droplets that undergo Brownian motion, and once they collide into neighbors, they coalesce into larger droplets that then sort of diffuse slower because of the 1 over r scaling of the diffusion. Okay, so it turns out that if you want to recover the experimental observation, you need to include a coalescence probability that is less than 1. So I guess the thing to say is that the sort of anisotropic nature of the particle sort of inspired us to think maybe droplets collide, but they don't necessarily coalesce. If you include this kind of prefactor here, it turns out it scales linearly with the coalescence probability, and the simulations we can recover the experimental observation as well. And so really the significance here is that we confirm not only that the mechanism is sort of what we expected, that the growth is limited by Brownian motion and coalescence, but with this finite probability. And so the thing we're thinking about now is, where does this 10 to the minus 2 probability come from? Something we didn't include here at all is electrostatic interactions. And we also did not include sort of the shape of the particle, the fact that there are only 4 relatively small sticky patches. And the last thing to say is that because of the flexibility of these particles, it's possible that nanostars on the surface of the droplet may be fully connected. You may not have, free bonds, every 10 nanometers, for example.
Just a quick question here.
Sure.
So I don't so as you mentioned, these are liquids. Do you consider there might be possibility of like depletion interactions between the liquid droplets or any sort of that?
No, we don't include depletion interactions. I think the answer is because of the mesh size of the particles. And maybe I'm not thinking in the right way, but I would imagine that if you had a depleting interaction, you would need a particle that has sort of a different occupied volume inside and outside the droplets. And so I'm not sure, maybe I'm not sure how to think about it correctly.
Okay, thank you.
Yeah. Okay. So this is all for deep quench sort of growth dynamics, but we can also perform experiments at different temperatures, and we can measure the delay time now for a constant nanostar concentration at different temperatures approaching the melting temperature. And what we see is that if the quench is deep, sort of like we expected, the growth will be limited by transport mechanisms, like I mentioned previously, so it'll be relatively constant. But then as you approach melting, the delay time to see droplet appearance on the microscope seems to diverge. And so we wrote down a very simple model that depends on sort of a classical nucleation. This increase in delay time here was sort of evidence of a nucleation barrier developing. And so we combine essentially this classical nucleation picture with the sort of the van der Waals picture of surface tension, because we don't have direct measurements of the temperature dependence of the surface tension. But what we do have is very good theoretical understanding of the equilibrium phase diagram of DNA nanostars. That is this wartime patchy colloid picture. And so we can use that to evaluate the free energy density and the difference in concentration as a function of temperature that can then give us an estimate of the surface tension. And so then we get a functional form here for the delay time, and we see that it fits pretty nicely, assuming that we have a mean field form of the surface tension here. And so really what's the value in doing this kind of thing is now that we can perform experiments at different temperatures and concentrations, we can estimate not only the equilibrium binodal line by the melting temperature here, but we can also distinguish the growth dynamics for all points in the phase diagram with this binodal line here. So the lines here are from the Wertheim theory, and the blue diamonds correspond to the estimates of the melting temperature, and the light blue circles correspond to estimates of the spinodal temperature. That is where the nucleation barrier is roughly kT.
Sam, can I ask a quick question over here with regard to the mean field prediction from classical nucleation theory? There is a prefactor in front of the the e to the delta G by kT. So the 1 over kappa, which sets the time scale, what do you assume about that?
So this is one of the fit parameters. I guess what we assume is that it is temperature independent.
Okay.
And the reason we do that is because we assume this is a transport sort of prefactor. In the absence of a nucleation barrier, what determines the growth? And that's essentially what we did here, right? So we have if I bring back this prefactor here, it sort of scales with the concentration.
Yes, but there is a kT there.
Yes, you're right.
It's not exponential.
Right. But that is, absolute temperature, not relative temperature to the critical point.
Yes. Okay.
Yeah, that's right. So yes, there's a slight dependence there, but this is 5 or 10 degrees.
Yes. So the assumption is that the primary dependence is all through the exponential, and there is no singularity, or no strong enough singularity in the transport coefficient.
Yeah, exactly. That's right.
Okay. And then you use the same fit parameter for the rest of the phase diagram. Is that correct?
Yeah, exactly.
Okay. Sorry, continue.
Okay. Yeah, ready?
There was one more question in the chat from Dan. In the simulation, so you mentioned there was this probability of coalescence, which is 0.01. So what happens the other 99% times? Do the droplets pass through each other with no interaction?
Yeah, that's right. So I sort of swept some of the details under the rug here, where I just say probability P naught, but because there are a distribution of droplet sizes, we have to be a little bit more careful about what we mean by an encounter. And so essentially the longest time scale here is this smaller droplet of a pairwise interaction, the time for a smaller droplet to diffuse through the diameter of the larger droplet. And so that's sort of the total encounter time. And then this P naught is normalized by that. But yes, you're right, that they diffuse through each other with no interaction. Of course, it's not quite right, but that would require a little bit more careful treatment, I guess. Yeah.
So I have a quick question about the surface tension. If you in the experiments, if you can measure the coalescence time, that can be used to back out like a ratio of the surface tension and the viscosity.
Yes.
So have you tried that, or?
So we've done it. So we've done it at a constant temperature in this deep quench regime, I would say.
Okay.
But really what we care about is the temperature dependence. And I'm sorry I don't have a backup slide here, but this sort of dependence difference in the concentration, sort of the uniqueness of the nanostar phase diagram is that the dense phase concentration is temperature independent for the most part. If I can go back to my phase diagram here. If you think about like protein phase separation, this just keeps getting denser and denser as you lower more into the coexistence regime. But because essentially it's a fully connected network after only a few degrees below critical, there's no energy gain from densifying the system. So this is essentially just as dense as the system can get. What we were interested in is how to model it near critical. And yes, you're right. Of course, we could do experiments, but it's not only one experiment as a function of temperature, these coalescence times, but it's actually two, because the coalescence only gives you the ratio of the surface tension to viscosity. And so we'd have to do a different experiment, like a FRAP or something like that, to estimate the viscosity, or just bulk rheology would also work. Yeah. So yes, the answer is we can do it, but given that it was two, we decided to think about it before doing the experiment in this case.
Mm-hmm. And could you remind the audience where the T cube relation comes from? Is it just an equation of state, or?
I believe it's a series expansion of the chemical potential. And this is, in a mean field sense, the first term that survives.
I see, like a virial expansion.
Yeah, that's right. That's right. And so if you do the Ising treatment, I believe it's not C cubed, but you get 3.8 or something like that, which we also considered.
Thank you.
Yep. Okay.
There's another question from the chatting from Christian. Do you have an estimate for the critical radius? Is it much smaller than your point spread function? I'm wondering if growth coalescence happens during what you call the delay time.
Yes, certainly. This is an important point and maybe I'll just conclude on this point before we move on, but we don't need access to the smallest length scales to estimate sort of nucleation-mediated growth as opposed to spontaneous growth. Because you're right, certainly even in these regimes that we can resolve, the critical radii are still only 5 to 10 nanostar particle diameters, which is less than the point spread function of the microscope. But essentially, because we have complete understanding of the transport mechanisms, that is the deep quench growth and the nucleation barrier, we can estimate the spinodal line from just micron scale dynamics. That's sort of the idea. Okay, so let's move on before I run out of time. Okay, and so the next sort of short thing I want to say is that we can also not only look at the assembly of just nanostar droplets, but we can start to control things like their size by introducing DNA surfactants. And so this is what we do exactly. We make a very long, quote unquote, surfactant, 400 base pairs by PCR. The idea here is this long, stiff, double-stranded DNA does not want to be confined inside the nanostar meshwork. So it is preferentially excluded, but because there's a binding sequence on this long DNA, it would like to localize to the surface. And essentially what we do here is we mix different ratios of surfactant to nanostars, and we can see control of the droplet size. So relative to just nanostars by themselves, which undergo unbounded growth, like I told you about before, if you start to insert a surfactant, these long DNA molecules can stabilize the droplets. And we were sort of inspired by dispersion polymerization of colloidal synthesis in that the ratio of the surfactant of the nanostars essentially determines the size. Sort of when you cover the surface with a high enough density of surfactant, that's what determines the size. And so this is like the quantum dot synthesis that was just given the Nobel Prize recently. But what I want to emphasize here is now we have complete understanding of the nucleation and surfactant stabilization of these nanostar droplets that really implies that we have the control needed to make highly monodisperse submicron droplets. So here I've shown you micron scale droplets, but if you increase the surfactant concentration here, you can decrease the droplet size into the 100 nanometer range that is important for certain applications like drug delivery, for example. Okay, so I just want to end that section there before I move on. So now I'm more interested not in the condensation dynamics, but in the spatial patterning. So I want to show this movie one more time where we form droplets by quenching into the coexistence regime. And now what determines sort of the arrangement of these patterns? Okay, so this is a thermodynamic process. So this system looks random to me. But so, okay, in an equilibrium sense, what determines the mesoscale structure is essentially the second law of thermodynamics tells you want to minimize the free energy. And essentially, there are only two structures that can result. You either maximize the entropy here on the left and you get a random arrangement of particles, or you bind to lots of neighbors and it turns, because of crystalline symmetries, you can bind to lots of neighbors and then minimize the free energy that way, and you end up with a crystalline solid. Of course, out of equilibrium, the entropy is not well defined, so people are thinking about how to characterize structures in an out of equilibrium environment. And one way to do that, which was formulated, roughly 20 years ago or so, was to characterize these structures by their long-range order, so-called hyperuniformity. So here I'm going to formulate this long-range order in the form of a density-density correlation function. So a random system here has density fluctuations on all scales. You can sort of see it here where there are regions of high-density clusters, also regions of no particles, whereas for the ordered system, there is no density fluctuations up to the first Bragg peak. So there are no, gaps here. What they found in formulating sort of the mesoscale structure in this way is a new class of systems, so-called disordered hyperuniform, that have long-range order like the crystalline structures, but also local isotropy in that this density correlation function here looks the same in every direction. And so this power law here of the decay of the density fluctuations has fundamental importance because this alpha encodes the organizational mechanisms of the system, particularly in non-equilibrium systems. For example, dynamical phase transitions and random close packing. But the one I want to focus on today, of course, is biomolecular condensates. And so essentially what we do here is we're going to measure this density-density correlation function of these images by just taking the power spectrum of the image itself. And what we do is we're plotting now the density correlation as a function of Q, and we find that the density fluctuations are heavily suppressed relative to what we would have expected for a random system, let's say. So what's happening here is that droplets are forming in solution, they're undergoing Brownian motion and coalescence, they're growing, and then eventually when they reach a size that is large enough, they sediment to the bottom of the container, and we measure these 2D arrays. So we thought maybe the sedimentation is driving this kind of ordering, but we can do the same experiment with colloids and we don't see this effect. So it really suggested to us that it's something about the phase separation that is driving this long-range hyperuniformity. So then I turned to simulation. We did a very simple Cahn-Hilliard simulation, and we can calculate the same structure from these patterns of droplets. And we see we're in the right direction, that the density fluctuations are heavily suppressed. And it turns out the power law here is even stronger than we expected. And that's because the droplets here in the simulation do not contain thermal fluctuations. So there's no Brownian motion in coalescence mechanism going on in the simulation. What we see is Ostwald ripening. So I'm certainly not a skilled enough simulator to include thermal fluctuations. And so the simplest thing I thought to do is I thought to make a pattern. And because the Brownian motion should be self-similar, it should be the same to just, instead of giving the particles random displacements throughout the simulation, I'll just give them all one big random kick at the end and see what happens. And so that's what we do is we take these droplets, we locate them and size them, and we give them random displacements according to Stokesian diffusion. And this is all it takes to match the experimentally observed structure. And essentially the magnitude of that displacement is the only free parameter here that tells you. And so we see the same power law, and then to match the prefactor, you have to pick the right random displacement magnitude. Okay, so you don't have to do this with nanostars. You can do this with a different kind of phase separating system. So we did this with peptides and nucleotides, where the microstructure of these droplets is very different. The phase separation mechanism has to do with entangled polymers and electrostatic interactions, but the patterns are the same. So we see the same hyperuniformity here. So I just want to emphasize that this is really a general feature of phase-separated systems that have sort of Brownian motion or noise large enough to disrupt the structures here. Okay, so if you have a phase-separating system and you want to look at the long-range order, all you have to do is look at the images of the droplet patterns, and maybe you'll see something like this. Okay, so the reason to use nanostar droplets is that now we can start to engineer different types of patterns because we have complete control over the nanoscale structure. And so here we've designed two different particles with two different sticky ends, and the idea here is that they will phase separate into different droplets because there's lots of energy gained between binding A, but no energy by design, essentially, for A to bind B. And so then you can start to think about, can we have these kind of composite hyperuniform structures? And the reason I think of this is because one of the first examples in living systems of what you would call hyperuniformity is the arrangement of cones in the chicken retina. It was observed that this kind of structure, there are 5 different types of cones in the chicken retina. They form this disordered pattern that is what they call multi-hyperuniform. So it's not enough that just the red ones themselves are hyperuniform, but all of them together are hyperuniform as well. So that's what they call them, multi-hyperuniform. Okay, so we can do the same quench into the coexistence regime, and we see the patterns of droplets form, and you can see that they start to form these different droplets that are now sort of interpenetrated patterns here. You can see they don't look random right away because the pink ones look like they're surrounded only by yellow and vice versa. So now we can characterize the structure in the same way. Just for the yellow ones, we see the same decay in the density fluctuations like we saw before. It's like it doesn't care about the other component in the system. The same is true for the pink droplets. However, if you include now both of them in treating this image like a grayscale image, the sum is random. And it looks like the long-range density fluctuations look like that of just equilibrium hard disks, so just including excluded volume interactions. This kind of result really drives home the sort of fundamental point that specific interactions that drive the phase separation are really responsible for the long-range hyperuniformity. But you also might imagine that there are some interesting applications you could think about for this kind of pattern. For example, biomolecular transport, where you'd imagine droplets that interact with a specific pattern, either the yellow or the pink, would have very different transport properties than a biomolecule that has a non-specific interaction to these droplets. And so that, yes.
Can I ask a question over here?
Yeah.
So you have these so if I understood correctly, the two components of the system, which you show in purple and yellow, are both individually, separately hyperuniform. So for them, you just take the intensities of the specific color channels. Is that correct?
Yes, that's right.
And then when you combine them, I'm guessing you're just adding the intensities of both, and then you measure the correlator that saturates at small q.
Yeah, that's right.
Okay. I'm curious if you have thought about a connection to an absorbing phase transition in this context then, because there are some recent results from my kids group that suggest, at least in a particular model of an absorbing phase transition, they have a very similar situation wherein you have two densities that are individually hyperuniform, but the combination is not. And so which requires then some sort of very delicate cancelling of the specific scalings and the prefactors between the two densities. Because presumably if you added the purple and the yellow density with different prefactors, you won't necessarily get this saturation. It'll only be for a specific combination that they will actually cancel out their sort of vanishing scale below Q. And at least for some class of absorbing phase transitions, systems with absorbing phase transitions, this seems to be a situation. So I'm curious if there is a way to think about the system wherein there is perhaps some version of an absorbing state.
Yeah, no, it's interesting. I don't know. Certainly an absorbing state doesn't really come to mind to me right now, I guess, except for one depending on depositing the droplets on a surface. The interesting thing is that the converse is sort of easy, that you take two random patterns and you can get a hyperuniform one out of it, because then you just start from a hyperuniform pattern and then you just randomly select two different subsets, and those will certainly be random, right? This one is really funny in that if I flash back to the crystalline picture I showed you before, and I gave all those particles sort of a small displacement, you would get this Q squared decay of the density fluctuations. What's really funny is to take two ordered systems without density fluctuations and add them up and to get something with density fluctuations. So it's really, I think, a little bit more subtle than sort of I had in mind when I first took this data, certainly. And so we're still, of course, thinking about this. From a mathematical perspective, you know that there are certainly cross terms in the correlation. The cross correlations don't necessarily need to decay to zero. And if your sort of self correlations both go to zero, that doesn't necessarily imply it. But there are conservation rules in that you can't remove or destroy mass. So there's something a little bit funny here.
Yeah, and I think having a situation like this will make a prediction for the cross correlations, that things have to be sufficiently anti-correlated. Your purple
that's right.
Will have to have some specific forms of anti-correlation to get this result. So anyway, it was just a thought that there may be an interesting connection to absorbing states.
Okay. Yeah, no, it's certainly something we're very interested in. Okay. The last thing I want to say about this point is I sort of mentioned a possible application for biomolecular transport, but we've also done some experiments where you can connect these droplets with these sort of surfactant particles that contain sticky ends from both of them. And then you can get these patterns of adhered droplets that form these percolated strings. And so this, you might imagine that this would have interesting sort of mechanical signatures of stress transport, not just biochemical transport, for example. Okay, so I think I'm running out of time, so maybe let me go quickly to the last one, and then we can discuss maybe more at the end. Okay, so the motivation for this last one results from this picture that I showed at the beginning as the nucleolus using phase-separated droplets to regulate the genome. So it's known that transcribed RNA participates in these kinds of liquid-liquid phase separated droplets, but also the thing I'm not showing you with this picture is that the genome is all in here as well, and the droplets condense around the genome. And so you might imagine this kind of control network where transcription can turn on a phase separation process that then regulates the initial transcription process by phase separating around certain parts of the genome. And so in order to develop a model system like this, we're turning to in vitro transcription, where a polymerase binds a template and then makes a copy of single-stranded RNA and coupling that to a DNA nanostar. And so we changed the design a little bit so that the polarity of the DNA hybridization is obeyed, where the synthesized RNA linker is now, what binds RNA or binds the DNA nanostars. And so, sorry, sticky clicker today. And so what we essentially do is we design these sequences at the end to be orthogonal such that there is no phase separation in the absence of RNA. And then as we synthesize these single-stranded linkers, they can bind the nanostars. So the first thing we do is we run a transcription reaction. We purify the transcribed RNA, and then we mix it back with nanostars in certain stoichiometries, and we find exactly what you would expect from an equilibrium phase separation process where you add more RNA given a constant nanostar concentration, and you get more droplets and larger droplets. And so we can quantify the total condensed volume from these images, and we find exactly the lever rule like you would expect. Except it's slightly different here because it's a two-component system where it's linear for low amounts of transcribed RNA. And once you saturate all the nanostar bonds, you get a constant afterwards. So now really what we're interested in is doing this together, forming the droplets and transcribing the RNA together. And so now we're going to do it in situ. And I'm showing you now similar images where there's different concentrations of RNA being transcribed. It's increasing as a function of time, but droplets only form much later than we would have expected for an near equilibrium process. And so if you plot again now this total condensed volume as a function of transcribed RNA for the transcription process, there's this significant delay time that really is emphasized in this nonlinear response of the transcription or of the phase separation as a response to the transcription process. And so here the droplets are saturated as well, but at later times. Okay, so now we can make droplets with an in vitro transcription reaction, but can we think about regulating this reaction with the phase transition? And so the way we're going to do it is by gene polymerase partitioning. So really what this in vitro transcription reaction depends on is the binding of RNA polymerase to a template, and then the polymerase zips along synthesizing the RNA. But if we separate the template from the polymerase, then they can't bind, they can't synthesize RNA. And so this is the mechanism. If you just have a uniform mixture, there is no reason you would think to do this because there is just a homogeneous bag of biomolecular components, but now we just made droplets. We have different biochemical environments, and so now we can think about engineering separation in that way. And so what we do first is we just look at, for an inert template, just a double-stranded template, this one is short enough so that it just goes inside the droplets. There are droplets here in the orange image, but the polymerase, essentially because it's quite big, it's on the order of the mesh size of the network, the polymerase is slightly excluded from the droplets. However, because the template is made of DNA, we can just include a different binding sequence on it. And so we include a nanostar binding sequence onto the template, and it goes into the droplets at a very high ratio here, 6:1 or so. So this is sort of what we have in mind, that we we sequester the polymerase from the template by putting the template inside droplets while excluding the polymerase. And so now we can plot the RNA concentration as a function of time again, and we see a factor of 3 or so slower reaction rate from the transcription reaction. So really, even though the droplets only take up about a 5 or less percent of the condensed volume, or sorry, the total volume of the system, it's enough to suppress the transcription reaction by a meaningful amount. And we can also monitor the condensation dynamics, like I showed you before. And the condensation dynamics of this partition template is much slower than the inert template case. So if you have template going into droplets, you see a very strong and sort of qualitatively different dynamics of the condensation process. And so that's where I want to stop for now. I just want to summarize by saying we're really thinking about these kinds of enzymatically driven phase separation processes as a way to control structure and reactions. So not only in a near equilibrium case where I told you about, we have a nice understanding of the droplet growth mechanisms of these nanostars, and also the surfactant stabilization for size control, but also the hyperuniform patterning as really a general mechanism as driven by phase separation and Brownian motion. But when we start to couple these phase separation processes to driven enzymatic reactions, we see non-equilibrium condensation dynamics show up sort of right away. And then we can start to engineer these inhibition control loops by spatial partitioning of the components. And so we're thinking about these in these sort of spatially patterned enzymatic bioreactors, something like a two-dimensional tissue where the reaction diffusion dynamics is coupled to phase separation processes. Okay, so let me end there. And thank you for your attention.
Okay, thank you, Sam, for this excellent talk. And now questions are welcome from the audience. Should I end recording now? No? Okay, just keep going.
Yeah, we usually record the first few minutes of the questions around the hour. We sometimes stop the recording there to have a more informal question session, but I don't think there's any strict rule about that. So we can keep it going for now.
Okay, so we can just keep going. So people can unmute themselves to ask questions or to type the questions into the chat box, and I will read them. So first, there are 2 questions in the chatting box. First from Dan, in the part that you do the Cohen-Hilliard phase field simulation, if you do know if adding noise to the phase field as opposed to the droplets as independent entities also recovers the lower power law?
Right. So, yeah, this is a good question. I think the answer is yes. In that we did these kind of simulations sort of after the paper was in a complete form, and we recovered this kind of noise, but I think it's in maybe a more trivial way in that the sort of the density correlations of the noise has this q to the -2 scaling, and so you just divide the Cohen-Hilliard q to the 4 scaling with q to the -2, and you get q squared. So it worked in that sense, but the phenomenological part of the droplets didn't really undergo that much Brownian motion. And when we added enough noise to make the droplets undergo Brownian motion, they also dissolved. And so I think there's sort of the stability of the solutions here is sort of a question of how do you add enough noise to let the droplets undergo Brownian motion while also having a high enough surface tension to keep them stable. Because the other thing we don't see is surface fluctuations, which you would certainly expect to see if you have noise in the simulation. And the other thing, I guess, to say fundamentally the model does not consider is that there's a difference in viscosity between the inside and the outside of the phases. So the dense phase here is, orders of magnitude more dense. And so maybe we could do it if we think about sort of a phase-dependent diffusion coefficient or something like that, but we hadn't gone that far yet.
Yeah, there's a work from Tanaka in Japan modeling colloidal particles as high viscosity droplets within the fluid, and it sounds like you literally have that. So I'm curious if their method would recover this dynamics, because hydrodynamics seems like it would be important at the magnitude of noise you're talking about.
Yeah, it's a good point. Yeah, I think that would work. I think the challenge is sort of one of scale, because that might play an important role on like a local scale, but probably on the long enough length scales, sort of challenging or sort of practically in doing a simulation on a large enough system where you could measure the long range order. But I also think the hydrodynamic correlations sort of die out, and I imagine you would be more thinking about sort of the hyperuniformity. The rule of thumb I think about for hyperuniformity is you end up, all the local stuff dies out, and you end up with conservation laws like mass conservation or for example, center of mass conservation of the interactions. That kind of thing is what determines the long range order. Yeah.
Thanks.
In maybe in that same context, so there is also some old work from Tomita from 91, which uses very simple isotropy arguments for why the Cahn-Hilliard scaling is q to the fourth without noise. But and it has to do with how when you don't have noise, the initial condition sort of is remembered, at least in the second order correlator. But when you do add noise, then that overwhelms the second order, the q squared component, which is what you should see, which is, I think, consistent with what you observe. So, and yes, the issue of the mobility, I think, is very interesting. We're putting in phase-dependent mobilities will presumably can also affect the coarsening, which would be interesting given that you see conventional 1/3 scaling.
Yeah, that's right. That's right. Interesting. Yeah.
Another question from the chat is, in the last topic, how could you track RNA polymerase?
Okay. Yeah, sorry, I didn't mention how we did this. So you can use your favorite sort of biochemical sort of chemistry there. Essentially, what we did is we did a sort of a click chemistry where we tagged the polymerase with azide. Sorry, I'm misremembering the other end of that reaction right now. But yeah, there are many ways to do it. We've done it lots of different ways. But yeah, you just add a fluorophore by clicking this functionalized molecule to the polymerase. And so we sort of shotgun tag all the polymerase there. And to be clear, though, we did not run the reactions with the tagged polymerases. So all the data I showed was on untagged polymerase. We just tagged to show the sort of near equilibrium exclusion properties of the enzyme there.
Okay, so other questions?
I was also wondering, since these scaling laws can depend on dimensionality, and you have this sedimentation effect that takes things from 3 dimensions to 2, is there a crossover time there where you might get some sort of 2-ish, 3-ish dimensionality messing with your scaling laws?
So you're talking about the hyperuniformity, right?
Right.
Yeah. Okay. So we did simulations both in 2 dimensions and 3 dimensions, and you get the same q to the 4th scaling. And you can do the same thing with the random displacements. I think, essentially because it's a mean field model, you're going to get mean field exponents.
Sure.
Yeah. So the other thing to say is experimentally, we were worried about the sedimentation for a long time. And so we did lots of experiments at different chamber thicknesses. And so we did very thin chambers and very thick chambers, and you see the same exponent. So the height essentially determines the total sedimented volume. So you see different droplet sizes and spacings, but the exponent is the same. Yeah.
So I have a question about as your DNA nanostar is structured like that, so it seems that the interaction between the nanostars is anisotropic. So when they meet and match that direction, they can bind, and if not, they cannot. So, but it seems that you just treat them as colloidal particles. You can see lots of. So the results are in most cases consistent. So on what scale or what aspect do you think that these anisotropic things will play a role to make it different from colloidal systems? Do you have a...
Yeah, no, this is a good point. I mean, we're investigating things on scales that are typically much larger than the individual particle. And so I think where these anisotropic shape really comes in is in the nanoscale sort of microstructure. And it turns out not very much is known about the microscale structure of these droplets. So there have been some scattering studies done on this where they find interesting effects of sort of the anisotropic shape. I'll just mention briefly that the four-armed nanostar that we typically use is actually a funny one because it prefers this what's called a stacked X configuration, where it's a very planar structure, and of course that depends on salt conditions and things like that. But there are certainly microscale sort of interactions that we're not thinking about that might play a role in things like the mesh size of the network, because you do other things like you can do rheology on these systems. That's been done, and the mesh, they do find a sort of characteristic length scale that has to do with the particle size, but there's also another characteristic length scale that has like a cluster size associated with it. And it's sort of unclear what sort of microstructural interactions. So yeah, clusters on the order of tens of nanostars that show up in the mechanics. And so there might be some lightly adhered connected network on the scale of tens of nanostars that can be easily sort of disrupted, or I come from a sort of jamming background, so I think of these as you might have some force chains that are holding the whole thing together, and then once you break those, you can have larger scale rearrangements.
Yeah, well, since you mentioned jamming, that's a question I had in my mind. So is there a part of the phase diagram where it really forms like a glassy or rigidified network which remodels slowly?
Sure. Yeah, I don't know if I would call it another boundary. We haven't really looked so closely at that, but certainly you're right. You quench deeper and deeper into the coexistence regime, the viscosity increases with this the Arrhenius sort of factor that you would expect, right? And so that's been done in terms of the viscosity measurements. I don't think there's been another sort of glassy transition, but I think it's challenging already to investigate. But certainly we see, one of the first papers our lab published was a comparison of these sort of certain conditions where you see liquid droplets form and others where you see this percolated network that relaxes very slowly. Yeah.
Yeah, and I was wondering, you chose these nanostars which have a very a coordination number of 4, which is kind of special.
Yes, right.
I mean, is it possible at all to play with different structures and change this multivalency, and that will probably have a huge effect on everything. So I don't know if it's
that's right. Are you thinking about crystalline structures? I mean, one of the reasons people were looking at these long ago, actually, I found some old Ned Seaman papers where he drew exact structures that I'm working on today, as he was thinking, this is how we're going to make a diamond structure out of DNA, just with these 4-arm junctions. It turns out for Ned, those junctions were much too floppy to make a crystalline structure. But you sort of imagine that you lower the salt concentration enough, the highly charged DNA backbone might repel enough to give you sort of a diamond structure. But certainly there are other even liquid property dependence on the number of arms as well, right? So the 4 arms is sort of anomalously dense given some of these preferential cross-like structures, but a 3-arm structure is much less dense. And then eventually you get to a higher number, and that you get to a regime where you necessarily get sort of glassy systems because you have lots of bonds that can form to neighbors. So I think part of the physics of the liquidity part is you can't bind to lots of neighbors if you have a limited number of arms. And so the ground state really is a liquid in this case. And maybe I should have emphasized this more because of this limited valence effect.
Mm-hmm.
Yeah.
Thank you.
I have another question about in the last topic you said that the mesh size is smaller than the RNA polymerized, so that kind of the droplets help separate the DNA and the RNA polymerized. So in real systems in the nucleus, the DNA is not the star shape like what you have. So that condensation, that droplets. Is there any kind of mechanism, some mesh-like things, or those structures to help separate those things? Do you know anything about the real biological system?
So yes, I would say I'm certainly not an expert in that area, but the mechanism for biological condensates is almost always an electrostatic coacervation of proteins and RNA. So positively charged regions on disordered proteins, let's say, and the negatively charged single-stranded RNA. In that case, the microstructure is very compact relative to DNA nanostars. You're thinking about highly entangled proteins and RNAs. And so in that case, the density of material is much closer to 30-40% by volume compared to these open DNA structures, which are only 1% by volume, right? So in some sense, we were worried that we had to engineer something about the polymerase to be excluded because we knew we had all this open structure inside the droplets, but it turns out the polymerase really is just massive. And so even though our structure is quite open, it's unfavorable. Essentially, you can think about it like, what is the energy cost of opening a vacuole inside the liquid, the size of the polymerase? And you do that estimation given the surface tension of the droplets, and it's tens of kT, or maybe not tens, but given that it's only slightly excluded, it's certainly greater than kT. Yeah.
Okay. If there are no other questions, I'll end the recording now. And